Hi viewers, good evening. Welcome to the new session of Remain Pro. Today we will do some practice questions of the ongoing chapter of molecular basis of inheritance. The practicing question will be of the previously taught lectures in the previous videos. So we are going to start with some questions, okay? And later in the later part we'll start with elaborately with the DNA world, okay? Now this is the lecture number 3 and of molecular basis. So let us start with the first question. If complementary base pair, what is the complementary base pair? Complementary base pair. Complementary base pair. So here in the complementary base pair, nucleotide, how many nucleotides are there? It's 6. Okay. Base is also 6 in number. Nucleotide 6. Base is also 6 in number. Base pair. How many number of base pair? Bases are 2. Nitrogenous bases. Either it will be adenine A double bond T and G triple bond C. Base B 2. That is base divided by 2. That is the number of base pair. If 6 divided by 2 equals to 3. Okay. Now the glycosidic bond. How many number of glycosidic bond? The number of glycosidic bond 6. So equal to number of nucleotide. Equal to the number of nucleotide. Now phosphodiester bond. Phosphodiester bond. Number of bases. Base 2. Linear DNA. So number of bases, linear DNA 6 minus 2. These are the key facts of this chapter. Okay. Linear DNA is 6 minus 2 equals to 4. Okay. Linear DNA is 4. Now what happens? The hydrogen bond, number of H2 bond 7. So these are the key facts. Okay. Now in case there are 2000 base pair, if the question comes in 2000 base pair, how will it contains 4000 bases? 4000 base. 4000 minus 2 equals to 3998. So phosphodiester bond if DNA is linear. This is the number of phosphodiester bond. Okay. Now question, if 3 base pair present, if 3 base pair is present, so phosphodiester bond, if 3 base pair is present, phospho bond, if DNA is circular, pseudomonas, in case of pseudomonas, okay, phosphodiester bond in case of pseudomonas. So phosphodiester bond, PB bond, number of base equals to 6. Okay. Now the adenosine monophosphate, AMP, IMP, CMP, AMP, IMP, CMP. AMP, IMP, CMP. AMP double bonds with IMP. Sorry, TMP. It's not IMP, it's TMP. Okay. TMP double bonds with AMP. And CMP triple bonds with GMP. Okay, so have you got it? Now what happens? Hydrogen bond, H2 bond, A double bond T, 2 double, T double bond A, 2 double, G triple bond C, 3 triple, C triple bond G, 3 triple. Okay, now come to the next important part, structure of DNA. Structure of DNA. Structure of DNA. So, who has done the X-ray structure? Franklin and Wilkin. Scientists Franklin and Wilkin X-ray fall on DNA fiber. DNA 2. X-ray reflected pattern study. 3. X-ray diffraction pattern of DNA that is the X-ray crystallography. 4. DNA is helical in structure. But they cannot provide the proper DNA structure. 5. Two types of study help Watson and Crick. They gave the detailed structure of DNA. Detailed double helical structure of the DNA has been given by the Watson and Crick. Detailed double helical structure. Okay, given by Watson and Crick in 1953. Franklin and Wilkin and B. Chargaff rule. Franklin and Wilkin and the Chargaff rule has been given. By these two theory, we know the double helical structure and elaborate structure of the deoxyribonucleic acid. Now, from them, we came to know that DNA is made up of two polynucleotide chain. DNA is made up of to polynucleotide chain. Polynucleotide chain. Both chain are antiparallel. So we know that there are two chains. So one chain goes from this up to down and another chain from down to up. So in case one chain phosphate at one end and second chain OH group. So in a chain there are two. In one chain there is a phosphate group and on the other chain there is an hydroxyl group. Okay. Now come to the next part. Watson and Crick structure. This is the most important questionable part. Watson and Crick structure. B type DNA, B type of DNA, right-handed DNA, right-handed DNA, oblique clockwise. Length of DNA, helix length, helix length, pitch of DNA, is equal to number of base pair into distance between two base pair. Vertical rise, here the vertical rise is seen in between the two base pair. Vertical rise is seen between base pair. The third point given by them, 10 base pair into 3.4 angstrom, that is 0.34 nanometer. 34 angstrom oblique 3.4 nanometer. This is one turn of DNA, helix length, length of human DNA, number of base pair into distance between two base pair. So what happens? Number of base pair is length of human is 6.6 into 10 to the power 9 into 0.34 into 10 to the power minus 9 meter. 0.3 angstrom is equal to 10 to the power minus 101 angstrom is equals to 0.3 0.34 into 10 to the power minus 9 meter. Okay, equals to, it will come into 2.2 meter. So length of one DNA is 2.2 meter. Diameter of one DNA, 20 angstrom. Okay, distance between two chains are uniform because purine forms bond with pyrimidine and vice versa. Purine becomes bond with pyrimidine and vice versa. So in vice versa, the pyrimidine also makes bond with purine. Now, here complementary base pairing. Complementary base pairing. What is complementary base pairing? Unique property of DNA hallmark. Unique property of DNA hallmark. Okay. So complementary base pair is the unique property of DNA. Stability of DNA. In the later you will come to know that RNA can also be taken as the genetic material, but the stability of the DNA is further more better than the RNA. So we take DNA, we consider DNA as the genetic material. Okay. Stability of DNA. Stability of DNA. Hydrogen bond. Stability of DNA. Hydrogen bond. Hydrogen bonding between base pair, base stacking. One base pair above other. Okay. Backbone. Sugar, backbone, phosphate. So sugar and phosphate makes the backbone of the DNA. So sugar and phosphate makes the backbone of the DNA. Okay. Sugar and phosphate consist of the phosphodiester. It makes phosphodiester bond. Now base is projected inside. Base projected inside. Now what happens? Chargaff's rule. Chargaff's rule. Valid for dsDNA. Valid for dsDNA. A. Purine equals to pyrimidine. A plus G equals to C plus T. A plus G, if considered as by C plus T, will be 1. Okay, by cross multiplication. A plus G, that is adenine, guanine, purine by pyrimidine will always purine. The division of the purine and the pyrimidine will always be considered as 1. B. Purine plus pyrimidine ratio is specific for species. Ratio specific for species. Okay. Now, adenine is equal to thymine. Guanine is equal to cytosine. A double bond T, that is considered as 2 hydrogen bond, and G triple bond C considered as 3 hydrogen bond. Okay, got it. Now climax. What's the climax? If question, adenine is 40, then what will be the cytosine? Then what is the value of cytosine? Okay, so we know adenine plus guanine, adenine and guanine is equal. So if adenine and thymine is equal. So if adenine is 40, then thymine has also to be 40. Okay. If adenine is 40, then thymine is also 40. So total we know it is 100. And now here adenine took 40 and thymine took 40. So remaining will be equally divided between guanine and cytosine. Okay. So remaining, what is the remaining? If 100 minus 40 minus 40. So remaining is only 20. So 20 will be equally distributed between cytosine and guanine. So cytosine and guanine will contain each of 10 and 10. Okay. Then C equals to 10 and G equals also be 10. Got it? Got the point? Okay. Because adenine equals to thymine and cytosine equals to guanine. Okay. Note, take it as a note. 5 into 174 has 5386 base oblique nucleotide. It is given data from NCERT. Length equal to the number of bases. Okay. Lambda fudge 48502 base pair. DS linear DNA. DS linear DNA. E. coli 4.6 into 10 to the power 6 base pair. Haploid. It is haploid. Human diploid 6.6 into 10 to the power 9 base pair. So 46 chromosome. Human genome haploid set 23 chromosome. 3.3 into 10 to the power 9 base pair. Got it? Got my point? Okay. So what we have completed till today, it's of DNA world, and in the later videos, in the upcoming videos, I'll try to start with the Watson and Crick model, the explanation given by the Watson and Crick, and later we'll go to the further topics of RNA. Okay. So till today, just revise it, and very soon this week the Neet application will be started. The registration for the Neet UG will start. So all the best for it, and good night.